All right, so let's keep talking about the cell membrane. So we were talking earlier about the basic structure of the membrane, phospholipid bilayer with proteins embedded within it that allow things to go in and out of the cell, that recognize things, all sorts of functions of those proteins. Definitely make sure you're comfortable with looking at those functions and recognizing function to a particular type of protein. So we left off last lecture with the movement of molecules, how they can randomly move. Some of them will move through the membrane if they are small enough. Size is the key for the movement through the membrane for the majority of molecules. If they're small, they go through, but they're always moving from a high to low concentrated area. So if you have a bunch of little molecules inside the cell, some of them will diffuse out. If there's a bunch outside the cell, some of them will diffuse in. Doesn't need energy, it's just natural motion of molecules. So an example of diffusion is breathing right now. We breathe in oxygen. The oxygen goes all the way down into these little sacs called alveoli within our respiratory system. So we go into the lungs, the bronchioles, and it keeps branching, getting smaller and smaller. And at the alveoli, there's a high concentration of oxygen molecules, and they're tiny. So they diffuse through that membrane, through that tissue layer there, and they will go from, there's the pen, they'll go from here, and they'll diffuse through that membrane and wind up in the bloodstream where they get picked up by the red blood cells, and then they get carried throughout the body. Okay, so there's an example of diffusion. You don't have to think about it or do anything, it just happens. Now, when we have a membrane, an event known as osmosis can occur. Osmosis does not occur without a membrane. Okay, so osmosis. Osmosis is the movement of water across a cell membrane due to a large solute being on the other side. So, to start out with, for osmosis to occur, you have to have a membrane. Osmosis is the movement of the water across the membrane. The reason the water is moving is because the big molecule, what we call the solute, is too big to go across the membrane. So water does. Water's trying to balance it out and reach this point of equilibrium. Okay, so some terminology I want you guys to get comfortable with. Isotonic. Same concentration of solutes inside and outside of the cell. If you are in an isotonic environment, water goes in, water goes out of the cell, but there's no net gain of water when you're dealing with an isotonic environment. Contact solution, saline solution in the hospital, etc. If you're putting a cell into a hypotonic environment, you have a low concentration of solute outside of the cell. More water is going to go in the cell than it will come out. So the cell will have a gain of water in a hypotonic environment. The cell will swell up and fill with water. The third environment is what's called hypertonic. This is where you have a high concentration of solute outside of the cell. And if there's a high concentration of solute outside the cell, more water goes out than comes in. In a hypertonic environment, the cell loses water. It shrivels up. It shrinks. Okay, so rule of thumb, solutes suck. Wherever you have your solutes, that's where water is trying to go. What the water is trying to do is balance out the concentrations and reach a point of equilibrium. Can it give you an equal concentration outside compared to the concentration inside? Equilibrium is what the cell needs to be in. If the cell's swelling up because it's gaining water or shriveling because it's losing water, that can destroy the cell. It needs to stay in its nice isotonic condition or balanced environment. All right, so examples. Saline solution, whether it's for your contacts or in the hospital, that solution is isotonic to the human body. There's a slight amount of salt in that saline solution, so it's isotonic and balanced to the body's salt concentration. If we put an animal cell in different solutions, it responds in different ways. Isotonic environment, some water goes in, some water goes out, but you notice the cell's got this nice filled happy cell shape. Isotonic is a good, happy environment. If you put an animal cell in a hypotonic, you got all this water rushing in with very little water rushing out, and this is where the cell can pop. It's like a balloon that's overinflated, and you pop. All right, not a good thing. Official term, lysis, or bursting. It pops the cell. It swells it up so much it explodes. Now going into hypertonic, more water is coming out than goes in, your cell shrivels up. This is what happens if you are to drink salt water. I need water and I'm in the ocean, take a big drink. There's a higher concentration of salt outside of your body, outside of your cells, than in the liquid you just drank. That will suck out the water from your cells. They will shrivel up. They will go through what's called crenation, or you're basically dehydrating your cell and shriveling it up. So we definitely want to try to avoid putting our cells in hypotonic or hypertonic environments because of the damage that can occur. Now, putting a plant cell into this environment, a little bit different. Plants are always a little different. Isotonic environment, nothing, no big deal. So keep in mind the structure in the center of the plant cell. You guys remember the term for the big bag of water? Well, that's what makes plants a bit different. The bag of water and then the cell wall that surrounds the plant cell. Those two features cause plants to act a little differently in these different solutions. So if you're in an isotonic environment, plenty of water goes in, water comes out, plant's happy, everything's good. Hypotonic environment for the plant. Water goes in, fills up, and then this thing called turgor pressure develops, and it fills up, fills up, fills up, but it doesn't pop because the cell wall retains the rigidity and the strength and the structural support. Instead of popping, what will happen is the plants actually can pump out the excess water so they don't pop. Now hypertonic, they lose their water, they shrivel up, they go into plasmolysis, that's just the plant version of shriveling up, but because of that rigid cell wall, they don't completely collapse. The central part kind of shrivels up and shrinks down, but the cell wall still retains its rigid shape. So now my plant's unhappy because it shriveled up. It dehydrated, it dried up. Well, shoot, come back and water your plant. And then you inflate it, and when you inflate it, it goes back to being an isotonic type of condition. So you guys ever get plants, flowers, roses, whatever, and they wilt, and then you give them some water, and they perk back up? You're like, wow, it doesn't look like anything's happened here. That's because of their cell wall and that structural rigidity they have. So they're a lot more responsive to being able to recover from hypotonic or hypertonic conditions. They can survive a lot better than if we don't have the water and the animal cells rupture or are destroyed. So, okay, so water's moving during osmosis. The key, again, I can't stress it enough, the key here is your substance is too big to go through the membrane. You're solute, and that's what causes the water to move. Osmosis does not occur if the molecules are small, and diffusion can occur. Now, sometimes cells need certain things in them that are a little too big, and they don't want to trigger or flip over and go into osmosis, so then a thing called facilitated transport can occur. This is when a special carrier protein, so remember our proteins, the carrier protein. Carrier protein will selectively move the molecule through the membrane. Kind of grabs it, pulls it in, kind of squeezes it, wiggles it, pushes it, and then it's inside the cell. So it's a way to get some solutes in that are just a little too big to go through the normal channel protein. So that's with carrier proteins. In an event we call facilitated diffusion. Now, here's the cool thing. Exocytosis. Okay, so with exocytosis, this is when the cell spits out a large substance. It says, I gotta get rid of this. It's too big, and I need to remove it from myself. So let's say it built a protein. Again, what are the protein factories? Or the cell built fats. What are the fat factories? It built something and it needs to get rid of it, but the substance is too large for diffusion and it's too large for that carrier protein. So hey, let's try this route. Let's put it into a bag. Let's put it into a phospholipid membrane. There's our membrane. There's our bilayer. That's just the bag, or what we call the vesicle. Let's push that into the actual plasma membrane. And because these are all phospholipids, and these are phospholipids, they literally can blend together and fuse together, and then it opens itself up, and those big molecules are now on the outside of the cell. So it's excretion, or expelling a large substance from the cell when we talk about exocytosis. Now the exact opposite is what this amoeba is doing here. So here's our amoeba, right there. This is the amoeba. And the amoeba is hunting, it's feeding, it's eating, it's saying, I want to get me that paramecium. And there's a paramecium. It is attacking and it's going to eat it. So it's going to bring that paramecium into its body through the process of endocytosis. Okay, endocytosis. Bring it into my body. It's too big to go through my membrane with diffusion. It's too large to go through my membrane with facilitated transport, but I really still want to get this stuff in. I'm going to do it through the process of endocytosis. So what we'll talk about in the next lecture is how this works. There's 3 different types of endocytosis, 3 different ways that the cell can engulf large substances that normally wouldn't go through the cell membrane.